Hey guys, welcome to Unit 4, Topic 1, PowerPoint 9, looking at mutations. I apologise that I've been so behind on making these. It's amazing how much less time you have when all of your classes are back from COVID-19. So let's go through and have a look at mutations. Okay, now this section covers two different parts of the textbook because it's kind of separated into mutations that happen in the gene, which is that section 5.4, versus mutations that happen across the chromosome, and that's on 7.1. So just in case you're confused where those pages are coming from. There's quite a number of things that you need to know from this lesson. The first is identifying how mutations in genes and chromosomes can result from errors across three different examples. The first is in DNA replication, and that's looking at point and frameshift mutations, which is just happening at a singular gene. The second is errors that happen during cell division, and this is called non-disjunction. The third is damage by mutagens, so looking at how different things in the environment can cause actual damage physically to the DNA. So we're going to look at UV radiation, heat, chemicals, and those sorts of things. Then from what you've learned about non-disjunction, you need to explain how that leads to aneuploidy, which is where we have incorrect numbers of chromosomes. You need to use a human karyotype, that's the chromosomal layout, to identify changes in chromosome number, and therefore from that predicting a genetic disorder from that. And then you need to describe how inherited mutations can alter variations in the genotypes of offsprings. So quite a lot there, quite detailed, and as you would have seen, there's a lot of tasks on OneNote that are associated with this. You might want to work through them all now. You might want to do some now and leave some for revision before you come to the exam. Okay, so when we talk about mutations, mutations are any change to the genetic information. And so as I said earlier, a mutation could involve just a very small part of the DNA. So it could even be a single gene looking at a base. So it doesn't need to be something involving large sections. It could just be one singular base on one gene. Some mutations involve multiple genes, so it might be a particular section of a chromosome, or sometimes it can affect the whole chromosome. So we're going to look at those three different examples. Some mutations don't affect the structure of the end protein that we get after transcription and translation, but sometimes they can still have effects on gene expression, changing the rate that occurs, or some can produce proteins that are non-functional. And in terms of how these mutations occur, sometimes it's random and the figure is around an error in every 1 million bases during replication, or it can happen due to exposure as an adult to things known as mutagens, things that are going to cause mutations to happen. Now, another sort of category that we can put mutation in is whether they are neutral, harmful, or beneficial. So neutral mutations don't have any effect on the survival. And so thinking about humans or the wild, this would just be a natural variation that's got no impact on survival. A harmful mutation would be something that's going to decrease the likelihood of survival. So it could be a trait that's going to make the animal slower, not able to hunt as efficiently, maybe have poor vision. For a human, it could be a disease trait, and that's going to affect survival. We also have beneficial mutations, and this could be something like allowing the animal to have better camouflage abilities to hide from predators and things like that. Now, when we talk mutations, especially on a gene like these, what you can see over here on the right, mutations happen, as I said, about once every million base pairs. And when we learnt about transcription and translation and replication, you learnt about some of the enzymes that come along and add the complementary bases. But what we didn't really talk about too much is that there's also enzymes that come along and check that there is no mismatching of the bases. Over here on the right, you can see this. So you can see that there's an incorrect base pairing of a G and a T, and that gets detected by special enzymes. And what those enzymes are capable of doing, Mr. Zell put up a YouTube video on the OneNote page, which is a really good one, looking at some of these repair mechanisms. But these enzymes can snip out pieces of DNA, remove them, and then our DNA polymerase or RNA polymerase can come along and replace that DNA to ensure that that error is fixed. And these mechanisms are super technical, and it's really cool how it happens, but this is why we don't often end up with mutations popping up too often, because our repairers do a pretty good job of that. Once a mutation is formed, though, it's established and it can't really be changed unless there's another mutation that comes along and makes some modification to that. Now, another type of grouping when we look at mutations is looking at somatic versus germline mutations. A somatic mutation occurs in the body cells. So this occurs in an adult, in an actual already living person, and affects only that individual. It can't be inherited. So that's things like if we get exposed to UV light and we develop a skin cancer, that's not going to be passed on to our offspring. That's something that has happened from adult exposure, and it's not going to be passed on. Germline mutations are those that affect the gametes. So they're going to impact the DNA that's in the sex cells. And those are going to be passed on to offspring because it actually affects the sex cells themselves. And so that's going to be something that can be passed on and was likely passed on to that person by their parents and so forth. As I said before, the mutations can be spontaneous or induced. And spontaneous is that natural errors that happen when the DNA needs to copy. That is often very rare, and it's when our enzymes haven't detected an incorrect base pairing, and sometimes it goes unnoticed or won't have any effect. It could occur on the introns where there's non-coding DNA, so we might not ever know that that's happened. The induced mutations are where we have mutagens that have caused a mutation to happen because of exposure. So that could be biological, chemical, or physical, which we're going to look at in the next slide. And that's going to cause that 1 in every million bases to increase and increase the likelihood of errors with that base pairing. So a mutagen is any factor that can induce a mutation. And if that mutation subsequently leads to cancer, it's called a carcinogen. So you often hear a carcinogenic substance, or and they're the ones that are going to lead to cancers or mutations leading to uncontrolled cell division. So we can have physical mutagens, so that's our radiation, so things like X-rays, alpha and beta rays, and our UV rays. We can have chemical exposure, so these are often things like industrial chemicals, pesticides, and they've recently come out as a carcinogen. And then we can also have biological mutagens, which we don't really think about, which is our bacteria and viruses influencing your DNA. Now, these are going to affect DNA in different ways. So they can cause bases to be replaced when they shouldn't be. It can cause errors with the bonding between bases or other parts of the DNA molecule. The effects can be quite different depending on the mutagen. So to summarize some of these, with our bacterial mutagens like viruses and bacteria, they're going to change the DNA of the human because they actually integrate their own DNA into our structure. And if you imagine that, that's sort of, you could end up with a random bit of DNA in amongst your DNA that's going to stop a sequence of bases from being read properly. Those chemical mutagens I spoke about, because the chemicals themselves have a chemical structure just like DNA, they can insert themselves into the DNA and cause strange bonding between the molecules, and that can cause the double helix to distort, stop the base pairing from happening. It can stop the polymerase enzymes from binding. So the chemical itself actually makes itself part of the DNA. And I've got a picture of that in the next slide. And then those physical mutagens with the different rays, the different types of radiation, that can physically cause breaks in the DNA. So it can cause parts of the molecule to not bind correctly anymore. And you'll see that in the next slide. So that's showing that radiation. This is this is ionizing radiation on the left, which is the X-rays. If you get an X-ray, you can't get them too often because X-rays can cause breaks in the DNA, and it's because of the actual energy that's contained within those rays. So you can see here that there's single strand breaks and double strand breaks. On the right is our non-ionizing radiation, and this is like our UV that's out in the sun, and that can create issues with the bonds between nucleotides. So you can kind of see that it's affecting this bond here, and it creates a kink in the DNA. And that's probably going to cause issues with the polymerase moving along and so forth. Now, heat is an interesting one because we know that heat causes denaturing of proteins, but we can also think of it as a mutagen because it causes the sugar and base bond to break, and the base can actually become detached from the nucleotide, and so it leaves a baseless site in the DNA strand. So if you kind of imagine that, you're going to have phosphates and sugars, but this bond here between the sugar and the base can break as a result of heat. And so you kind of can end up with bases floating off and not being connected to the backbone of the DNA anymore. And that can cause gaps and subsequent effects when we start looking at transcription and translation. Now let's start by looking at mutations from DNA replication. And this can also happen when we think about our transcription and translation and making of complementary strands of DNA or RNA. So back in lesson 2, we learnt that all the DNA has to copy during replication before the cell can divide. And that was what happens in interphase. And this could be looking at mitosis in the somatic body cells, or meiosis when we look at the sex cells. So that would affect the germline cells, the eggs and sperm. So the two different examples that we're going to discuss, which were mentioned in the syllabus, are point mutations, where we have one singular base involved, and frameshift mutations, still involving one base, but it affects all of the bases after that one base. So just like we can see on the left, a point mutation is where we have a base that's incorrect. So it could be one of those spontaneous errors, and you can see here that instead of a T, a C has accidentally gone in there. And we're going to look at what that could lead to. New day, and I'm here to continue the same PowerPoint. So in terms of point mutations, that's where we have one base that's changed, but that can have several different effects. The first is that we can have a silent mutation. So over to the right, if we have a look at what a normal triplet code would look like, so say it's TTC, and that's going to code for the amino acid lysine when we look at the table. With a silent mutation, we get one base change, so for example TTT, but if we look at the codon table, that still codes for lysine. So even though it's a point mutation where one base is changed, the amino acid that's made is the same. So that means it would go unnoticed and there wouldn't be any physical effect on the protein that's made. The second type is a missense mutation, which is where one base changes again. So instead of TTC, we have TCC, and this time it does produce a different amino acid. So it's now going to make an arginine instead of a lysine. And this means that the protein that gets made at the end is going to be dysfunctional in some way. So an example of that is sickle cell anemia, which you can read about, and we're going to speak about a little bit further. So in this case, it makes the protein not do its normal job correctly because one of its building blocks are incorrect. The third type of point mutation is a nonsense mutation, and this is where again one base changes. In this case, we've got an A instead of a T, but instead of coding for an amino acid, it codes for something different like a stop codon. So in this case, the protein is going to stop being produced, which could mean that it is very much incomplete. So in this case, the protein will be non-functional, and this is what happens in things like cystic fibrosis. Now, the second type of mutation that affects the gene at a gene level is the frameshift mutation. And these are a little bit more serious than the point mutations in some cases. And this is where we get an insertion or a deletion of one or more bases. This is worse because if we think about our triplet codes, having a look at this image here on the right, if this is our regular DNA sequence and we have, for example, an insertion, so see how this C base has slotted itself in between the original C and the T. This is going to affect every codon after that insertion. So instead of having CTG, CAG, GT, and so forth, it's going to mess up every triplet after that point. So we're going to have a CCT. And now instead of a CAG, it's going to be GCA, GGT. So everyone shifts up, and it's going to mean that every codon after that is incorrect. It works the same way with the deletion. So if you imagine, if I just get rid of this, if we came back up to our top one here and deleted this T, then instead of CTG, we're going to have CGC, AGG. So it works in the same way. And so these mutations often result in severe genetic diseases because that a whole gene can be affected by that. And there's some examples of that in the little table here for you to see. So those ones are looking at mutations at a gene level, so changing one base and altering a gene. These types of mutations now are going to be looking at errors when the cells divide. So this could be in mitosis, if we're talking about a somatic body cell, or in meiosis, which will affect the germline cells because it's going to affect the gametes, and that means that that will be passed on. If we talk about a chromosome, like we can look at whole chromosomes, which we'll look at in a minute, or really large chunks of chromosomes. And mutations that affect those big chunks of a chromosome are called block mutations. And there's a lot of them. There are duplication, deletion, inversion, insertion, and translocation. They all involve something happening to a large chunk of chromosome, but just in different ways. So I'll show you what that looks like now. Now, each of these figures have come from Pearson, spanning across page 328 to 329, where you can have a little bit more of a read about these. The first one at the top left is showing a duplication mutation. So this one here is the normal chromosome, and it's just marked 3 different regions, A, B, and C. And you can see that the C region has remained the same, but up here we have a duplication mutation where A and B have duplicated themselves. This isn't the worst thing because the genes are still there. So the transcription and translation can still occur. It's just been duplicated, which shouldn't happen. To the right here, this is a chromosomal deletion, and this is worse because we're now missing a section, and so we're not going to have that DNA to code for those genes. So the normal chromosome's got A, B, C, D, and E. The mutated chromosome is missing that B section. So that could be multiple genes. So more severe again than our point or frameshift mutations. Down here we have a chromosomal inversion, which is where you have a chunk that's removed and then swapped around and inserted in the wrong way. Normal chromosome A, B, C, D, and E. Abnormal or mutated chromosome, you can see that the A, B, C, and D have done a switch. They've reversed the direction. And again, that can be severe because we know that our DNA is read in a particular direction, so everything is going to be back to front. And that would cause a bit of a jumbled protein to form. The last one here is an insertion where you actually have a piece from one chromosome, A, B, C over here, swap over to this one and insert itself into an incorrect chromosome. So it's in the wrong position. So coming back to what we've learned about meiosis, this could or could not be inherited depending on the chance events of random assortment. Remember, the chromosomes align randomly, so different things could happen. With the situation on the left here, this would be ideal because we've got the chromosomes lining up at the center, and this is that deletion, that insertion mutation that we just looked at in the last figure, where one piece was taken from this chromosome and added to this one. These ones on the left are normal. Because of the way that they've aligned and divided, this gamete is still going to be normal because it has the full copy of the blue genes and a full copy of the red genes. And over here on the right, you've still got the blue genes and you've got the added part of the red gene as well as the existing red gene. So even though it's in the wrong place, all the daughter cells are still going to have a copy of every gene. So transcription and translation can still happen. On the right though, if they align in a different way, so this is the missing one and this is the inserted one, as they divide, the one on the left here is going to be missing that section, that red section that was taken from it, and the one on the right has two copies of that. So that's not so bad, but this is going to be bad because there's actually missing genes there. So that's just to give you some context of how it's a random chance whether sometimes these mutations even get noticed or passed on. This one here is a translocation mutation, where a whole segment of a chromosome gets swapped with another. So again, normal here on the left, and you've got these sections that have been swapped across the chromosomes. This can affect gene regulation. So it does mention things like cancers and things can come from this type of thing happening. So what we've looked at so far is looking at chunks of chromosomes that have mutations taking place. So it could be swapping, adding, deleting, and so forth. What can also happen is during cell division, the chromosomes can fail to separate correctly. So if you imagine when the chromosomes line up at the center during metaphase and the spindle forms and grabs onto the chromosomes, when they contract, they're supposed to take half to each side and each daughter cell gets a copy of that information. But sometimes non-disjunction can occur, which is where they do not separate. And so when it contracts, it might take more to one side and leave one side with less. And that can lead to abnormal numbers of chromosomes, which is called aneuploidy. This can happen in mitosis, where the sister chromatids don't separate properly. It can happen in meiosis at either meiosis 1, which is where the homologous pairs fail to separate. So thinking back to when we have our homologous pairs and then the spindle forms, and then it takes one to each side. So what can happen is that it takes both to one side and takes none to the other side. Or meiosis 2, which is after that's happened, the second division fails, where instead of taking a chromatid to each side, it takes the whole chromosome and leaves the other side with none. So I'll show you what that looks like now. This has come out of your textbook, so you can see the difference here. Meiosis 1 would be where the homologous pair didn't separate correctly, and instead of separating these two, it's taken them both to one side. And so we're going to end up with this cell having extra chromosomes and this one having not enough chromosomes. And then meiosis 2 will occur as normal. And so that means that these two gametes are going to have an extra chromosome, which is called trisomy, where you've got 3 copies instead of 2. And these ones have too few chromosomes. So this is the classic case of what happens in Down syndrome, where you have this occurring on the 21st chromosome, which is why it's called trisomy 21. In the second example here, meiosis 1 has occurred normally, and the daughter cells have divided normally in that first division, but in the second division, the sister chromatids have failed to separate correctly. And so this means that only these two are affected because this side happened normally. So again, we have one with too many, one with not enough, but these two are normal. So there's a 50-50 chance of the offspring having an abnormality in the number of chromosomes there. So that's non-disjunction. Now the syllabus asks you to know what a karyotype is and to be able to use it to make predictions about genetic diseases. So looking at things with errors in the chromosomes. So a karyotype is what you see in the image here, which is where you have a layout of an organism's chromosomes. And in the activities on the notebook, it goes through how this happens. So you can get this from taking DNA samples, performing some chemical analysis on them, and piecing together the homologous pairs. And what we can do is look for important things like changes in numbers of chromosomes, which would suggest things like that non-disjunction. We can easily identify the sex of the organism if there's the correct numbers of sex chromosomes and so forth. So we can use this to make predictions, which we're going to have a look at now. Now this one here is showing aneuploidy, which is that trisomy 21 Down syndrome example. And you can see this because you should be looking for pairs. And in this case, we have 3, which would suggest that there has been non-disjunction on that 21st chromosome, meaning we have an extra copy, which is going to be our trisomy. This one here is looking at some of the sex-linked disorders, which you can have a look at in the mutation activity. The one at the top here is called Klinefelter syndrome, and you can see that the abnormality is on the sex chromosome because it's number 23, and instead of having 2 X's or an X and a Y, there's actually 3 sex chromosomes. So it's often called XXY syndrome or Klinefelter syndrome. And this is where you're going to have a male because there is a Y chromosome that encodes for male-specific genes, but they are going to be infertile. They won't be able to have their own children. And there's going to be some other things going on like breast development because of having the extra X chromosome. This other example here is called X0 or X0 because there is only 1 copy of a sex chromosome. In this case, it's going to be a female because there is no second chromosome to encode for male traits, but the female will be infertile and often short-statured. And this is because there is only 1 copy, which comes with its complications. These are just some characteristics of different genetic conditions, and you can have a look at these. But the syllabus wants you to be able to look at those karyotypes and make some predictions about what's wrong. This is out of your textbook using a karyotype and coming up with what's going on. So the first question asks you to determine the sex of the individual, and to do that you need to go straight to chromosome number 23. We can see that we have an X and a Y, which means that it is going to be a male. The second part asks you to justify whether or not this is normal, a normal karyotype. So we're going to look at the chromosome pairs, and we can see that everyone exists in pairs except for pair 13. And if you look back at the previous table, that's Patau syndrome. You can have a look at that. So this is not a normal karyotype. We have a male with Patau syndrome. So you need to do some activities on this because you're really not going to be able to do a lot of these things until you really get in and have a go at it. So remember, you need to identify mutations in genes and chromosomes. So you need to understand point and frameshift mutations, non-disjunction, and how mutagens can affect DNA. You need to explain how we get aneuploidy from non-disjunction. You need to use karyotypes to identify changes in chromosome numbers and predict genetic disorders from given data, and describe how inherited mutations can alter variations in genotypes. Now, the syllabus states that even though you need to identify the mutations occurring and what they do, it says that you're not required to identify the effects of mutations, for example, silent, missense, and nonsense mutations. So you need to get how they happen, but not necessarily the effect that they have. It also says that recall of specific chemical mutagens is not required, but rather you should have an understanding that a large number of chemical mutagens are carcinogenic and interact directly with the DNA. It also says that for aneuploidy, the example could include trisomy 21. So I would take that as a good indication that you should understand how Down syndrome works. Now, there is a heap of review questions that you can do on this, but on the notebook, there's a stack of activities that would be really useful for you to do in your own time.